Hello, my name is Kristen Hope Wenzel, and today I want to discuss a study that looks at the invasive species, the Burmese python, and their involvement in transmission of mosquito-vectored pathogens. While most studies focus on their trophic impact, this one focuses on the more indirect effects that Burmese pythons have on the vector of transmission of disease due to their high predation and dramatic increase in population. So what are invasive species? How do we decide if something is invasive? While this is a controversial topic with a wide array of answers, we typically think of invasive species as non-native species that cause harm or damage to native life. They are often thought of as species that reduce diversity by competing with native organisms for limited resources. They often cause disruption in natural communities and ecological processes. In extreme cases, they can lead to extinction of native species. They tend to be organisms with high rates of reproduction, high dispersal rates, high genetic variability, broad diet, and short generation times. They can have severe impacts on human health by serving as carriers for human disease or being poisonous or caustic. So how do invasive species get into new environments? There are several different methods of transportation to new ecosystems. One way is from ships' ballast water that is carried from port to port. Organisms will also travel through wood, shipping pallets, and crates that are transported overseas. With smaller scale traveling, organisms can attach themselves to boats and their propellers to later be carried to new territories. And lastly, they can be introduced to new areas by humans through private ownership. In 2013, it was estimated that 19.4 million households owned some form of exotic pet. There are several cases where animals become too difficult to manage and will be released into non-native wild, where they proliferate and disperse. Burmese pythons are an invasive species that are a large-bodied snake native to Southeast Asia and prey primarily on vertebrates. They were most likely introduced to the Everglades National Park several decades ago by releases or escapes from private ownership. Sightings and removals of the pythons in the Everglades National Park were sporadic in the 1980s and 1990s, increasing drastically in the early 2000s. During this time, gut content analysis indicated that mammals accounted for 75% of their diet. Burmese pythons have vastly reduced mammal populations in the Everglades National Park. In 2012, it was found that most mammals had a decrease in population by around 90% and saw the complete loss of marsh rabbits, cottontail rabbits, and foxes in the area. Many of these mammals are ill-adapted to escape these type of predators, having never encountered them in the past. This shift in mammalian diversity leads to the increase in population of smaller vertebrates, altering the blood meal pattern of mosquitoes. There are many factors that go into why Burmese pythons have been so difficult to manage. Despite spending $10 billion on management techniques, the Burmese python still costs the Everglades National Park around $1 million a year in damages to environment and entrapment programs. Some of the actions taken to hunting down these Burmese pythons include capturing programs, using trained hunting dogs, and planting transmitter snakes that are tracked to popular mating spots. None of these has managed to be effective. One reason is that the Burmese python are very good at staying hidden and camouflaging to catch prey. There is also the ongoing issue of climate change that has allowed the pythons longer foraging seasons before going into brumation. It has also been found that Burmese pythons and many other large snakes are able to thermoregulate the temperature of the eggs at much lower temperatures than previously thought. The average female can produce anywhere from 50 to 100 eggs annually. While Florida has banned the sale of Burmese pythons, people continue to acquire them and release them into the wild when they become too big to manage. When looking at the environment, there are a few key events that have played a role in the Burmese python's proliferation. The first is Hurricane Andrew of 1992. It was reported that a large breeding facility of Burmese pythons was hit by the hurricane and aided in the dispersal of eggs and mature pythons. Another event was the deep freeze of 2010. While initially resulting in an estimated 40 to 90% field mortality, the pythons that did survive were more resilient and better at adapting to the cold weather. A study in 2018 performed by the University of Texas at Arlington looked to find if there was any genomic change in the pythons from before and after the freeze. By scanning regions of the Burmese python genome, they identified parts of the genome that changed significantly between the two time periods. This provided clear evidence of evolution occurring over a very short time scale in this population. While they expected to find genes in the region important for potential adaptation to cold, they found there was instead adaptation in the genes related to cell division, organ growth, and tissue development. They continued to find that the pythons had become better adapted to constant feeding and regulating their digestive physiology. They were found to have tissue morphology and gene expression that supports a more upregulated physiological state in fasted pythons. This means the pythons are getting better at being an invasive predator and constantly consuming prey. An example of their effects on mammal survival can be seen with a study conducted by Robert A. McCleary that tied the Burmese pythons to the rapid decline of marsh rabbits in the Everglades National Park over the last several decades. Marsh rabbits are particularly resilient in the Everglades National Park due to their ability to reproduce year-round, producing up to 6 litters of 3 to 5 young per year, and their persistence under considerable predation pressures occurring prior to the python arrival. With this in mind, there was a considerable loss of 77% of all marsh rabbits in the area, despite being a generally hardy species. These findings seem to contradict ecological theories that a single apex predator, aside from humans, could remove small, broadly dispersed, fecund generalist herbivores. McCleary's study took 95 adult rabbits, 80 of which survived the 10-day adjustment period, and found that out of 68 rabbit mortalities in the testing site, 77 were attributed to Burmese pythons, and only 1 mortality was linked to mammal predation. This contrasted greatly with their controls, in which 71% of mortalities were attributed to mammals, and only 3 mortalities were related to rattlesnakes. The loss of these mammals causes massive rearrangement of the Everglades National Park food web, which in turn can have unpredictable cascading effects. That, along with pythons' ability to switch to different prey and go long periods without food, make it difficult to regain the previous biodiversity. Without action, this massive reduction in mammals will remain unchanged and will most likely continue as the Burmese pythons continue to grow as a species. Having a diverse vertebrate community often offers as an ecological service to dilute the spillover of zoonotic pathogens to humans by decreasing vector contact with pathogen reservoirs. This is due in part to the difference in their susceptibility to infection and potential to transmit infection to other hosts. With more diversity, there is a decrease in abundance of reservoir hosts and prevention of them becoming widespread, reducing their effect and disease risk. This is especially true with rodents, which carry more zoonotic pathogens than other mammals and have more species that host zoonotic pathogens. The Everglades National Park is not the only region experiencing shifts in reservoir hosts and spread of disease. A current example of reservoir shifts in disease can be seen with the dramatic increase of Lyme disease in eastern North America. While the decrease in mammal diversity is largely due to deforestation, there are still great similarities in disease vectors when compared to the Everglades virus. The decrease in mammal diversity allows for the increased survival of smaller vertebrates. This works in favor of ticks carrying Lyme disease and allows greater survival and infection rates. Their current widely spreading host is the white-footed mouse. Due to climate changes and decrease in natural predators, the white-footed mouse is thriving in new areas they have not been seen in before. With this, they are also carrying the ticks with them and spreading their range as well. It is estimated that throughout the region of Virginia to Maine, at least half to sometimes up to 90% of the mice are infected with the Lyme bacteria. Humans are then being infected by coming into contact with spreading ticks or being bitten by the infected mice. While natural predators would typically eat infected mice and digest the bacteria, the lack of larger mammals allows the spread of mice to new areas. The CDC reports a near 300% increase of reported Lyme disease in the eastern region. With the decrease in vertebrate diversity in mind, the authors were looking to see if Burmese python's impact on the mammalian community has, in turn, affected the vectors of the zoonotic pathogen Everglades virus, or EVEV. EVEV is an arthropod-borne virus that is typically rare in humans. It can cause anything from fever and headaches to encephalitis, viral hemorrhagic fever, and neurological damage. More specifically, it is an alpha virus included in the Venezuelan equine encephalitis virus complex. The virus circulates among vector mosquitoes and is transmitted to the amplifying rodent. The mosquitoes in the amplifying rodent continue to spread EVEV to humans through their bite. It is a virus endemic to Florida, and the geographic range mirrors that of the mosquito Colixidaceae. Colixidaceae is the sole enzootic vector of EVEV and have been found to have a vector abundance that was dynamic across seasons and regions of the Everglades National Park. So why do we look specifically at Colixidaceae when there are 4 other species of Colix in the Everglades National Park? Well, for starters, the other 4 species of Colix feed primarily on birds and reptiles, which are not reservoir hosts of EVEV. Colixidaceae is one of the only species that remains abundant year-round, and particularly in dry seasons in the lower regions of the Everglades National Park. They have also been found to be highly efficient, having high transmission rates of EVEV to rodents. And lastly, there is rising concern that the introduction of Colix panicosa in Florida may work with the Colixidaceae to increase areas at risk for the Everglades virus. A study in the late 1970s found that the mosquito Colixidaceae was the sole enzootic vector of EVEV, and that 88 to 90% of its blood meals came from mammals, with half from rodents and the other half from medium to large-sized mammals. By the early 2000s, the loss of the majority of those mammals led to the mosquitoes relying more on the hispid cotton rat, Sigmodon hispidus, and the cotton mouse, Perimyscus gossypinus, which are reservoir hosts of Everglades virus. The authors decided to look at the host use of the EVEV vector Colixidaceae prior to python invasion with current host use patterns to investigate how an invasive apex predator may alter host vector contact. The authors started by measuring host use of the mosquitoes inside and outside the introduced range of the Burmese pythons using PCR-based blood meal analysis. They compared their results with data obtained prior to the introduction and rapid growth of the Burmese python in the Everglades. Blood-fed mosquitoes were sampled in the Everglades National Park and Vero Beach, using a range of 14 to 27 resting shelters per sampling day to target the Colixidaceae and their specialized resting behaviors. Everglades sampling was done at 8 locations, all occurring on hardwood hammocks of the sawgrass prairie. Sampling was conducted on 20 days, spread across 5 non-consecutive months, December 2015 to August 2016. In Vero Beach, sampling was done weekly from December 2015 to November 2016. This study analyzed blood meal samples using previously published PCR-based protocol, whereas previous studies used antibody-based methods to identify vertebrate source of mosquito blood meals. The chi-squared test of independence was used to compare historical and current distributions of total host blood meals, reservoir versus non-reservoir host blood meals in the Everglades, and seasonal patterns of reservoir versus non-reservoir host use. They then compared the abundance of Colixidaceae between the Everglades and Vero Beach using the Poisson regression modeling, which is a generalized linear model form of regression analysis. Analysis of the mosquitoes' blood meals from the Everglades displayed a dramatic shift in taxa-specific host use between the historic and current blood meal data. It also showed a significant shift towards feeding upon aggregated reservoir hosts, such as the cottonmouse and the hispid cotton rat. These findings were not related to seasonal shifts in hosts. Feedings on the hispid cotton rat, considered to be the most important EV-EV reservoir host, increased from 14.7% to 76.4% of overall blood meals. However, feedings upon deer, raccoon, and possum in the Everglades decreased greatly from 45.1% to just 0.8% of total hosts. There was interestingly a significant decrease in the use of the cottonmouse as a host as well. The study does not address or explain why the mosquitoes preferred the hispid cotton rat over the cottonmouse. The Shannon-Weiner Diversity Index also shows a decrease in diversity among hosts fed upon by 40.2% between historic and current studies. The mosquitoes were also more abundant in the Everglades than Vero Beach. On average, 24.5 times more females were collected per resting shelter in the Everglades than Vero Beach. The Colixidaceae from Vero Beach fed upon a wider diversity of mammal species when compared to those from the Everglades. Medium and large-sized mammals made up 37.5% of total hosts in Vero Beach. The eastern cottontail and the marsh rabbits made up 20.1% of overall blood meals, while deer, possum, and raccoon made up a combined 13.6%. The eastern wood rat, Neotoma floridana, made up 36.4% of overall blood meals and is not considered to be a reservoir host of EV-EV. In comparison, the hispid cotton rat made up only 18.1% of total blood meals. This data shows substantial shifts in the host use pattern of Colixidaceae have occurred in the Everglades. These shifts correspond with the decline of medium and large-sized mammals attributed to the establishment and rapid increase of Burmese pythons. The study highlights the indirect effects an invasive apex predator may have on the contact between vector and reservoir hosts of a vector-borne pathogen by restructuring the vertebrate community. It also shows that rather than expanding to non-mammal hosts, mosquitoes relied more heavily on small vertebrates that act as EV-EV reservoir hosts. However, there is still large debate whether greater host species richness decreases risk. It also should be noted that while it appears blood meal patterns have shifted over time, there is no data to compare populations of Colixidaceae and whether their numbers have changed in the Everglades. This means the authors cannot make substantiated inferences to the total number of infected vectors current day. Abundance of infected vectors is generally agreed upon as being the most accurate or reliable metric for quantifying pathogen prevalence and disease risk. Future studies are needed in order to determine pathogen prevalence and whether invasive apex predators increase risk of human exposure. So what does this all mean for big picture look at arboviruses? We know they require a vector arthropod and an amplifying vertebrate host. This study also suggests that another aspect of arboviruses is the biodiversity of the community in which they circulate. While it's obvious that global climate change and changes in human demography can alter the spread of disease, this study shows that the effect of invasive apex predators may also play a role in the shift in potential reservoir hosts and infectious vectors. With over 500 known arboviruses, 100 of which are capable of causing disease in humans, It is important that we develop a thorough understanding of how they are spread and the factors that may contribute to the emergence of new arboviruses, such as invasive species and destruction of biodiversity. So what can be done with this information? Well, firstly, it is important that we educate the public on the responsibilities of owning exotic pets and the proper care of them. It is important they understand the ramifications that invasive species have on not just the ecosystem, but also factors that affect them, like transmission of diseases. We also need to continue our efforts in attempting to reduce the Burmese python population. Many compare this invasion to that of Guam's brown tree snake. Guam is not only seeing a loss in biodiversity, but also a reduction in new growth of trees in response to their high rate of predation. These are effects we can only see down the line after the species has taken over the ecosystem. It is important that we track Burmese pythons and their trajectory in order to avoid them spreading into new territories. Lastly, further studies are needed on the Culex cetaceae and the Culex panicosa to see their effect on transmission of EVEV and potentially gather enough data to look at the abundance of infected vectors.